Okay, so hello, good afternoon. So we will start the last lecture in this semester, and I'm the guest lecturer for this week, and my name is Jiadong, and I'm a postdoc working with Kim An. So today's topic is deep learning solutions for single-cell RNA-seq data analysis, with a focus on probabilistic modeling based on SCVI. So we have a long title here, but essentially we're going to look at deep learning and its application to single-cell RNA-seq data analysis. Well, it is a relatively new field in single-cell applying deep learning techniques. But one of the most classic methods is SCVI, originally published in 2018. I chose this particular algorithm to focus on because it is widely cited. It is recognized, and it's more importantly, it's the SCVI library is a Python library, is still actively maintained. So, and on top of SCVI is a kind of deep learning model for single-cell RNA-seq. But on top of single-cell SCVI, we also have other the same team also built other tools for example, single-cell ATAC-seq and single-cell and site-seq, for example. So if you go to their website, you can see a range of tools for different purposes, all built in similar spirit as SCVI. So if I explain what SCVI is for you, then you should be able to easily understand how other tools in the same library works. So it's a very good example to for understanding deep learning techniques and also for understanding single-cell RNA-seq data. So this week we will discuss. First, we will look at a bit of history of deep learning. I call it history of deep learning prior to deep learning, because deep learning is a relatively new term. It was popularized around 2010, so around 12 years ago. But it's not something from out of nothing. Deep learning is the result of the result of the accumulation of many related modeling techniques from biology, from cognitive science, from computer science, from statistics. So it is really the result of the accumulation of development in various research fields. So I felt it's necessary to review some of its history, because deep learning is so topical nowadays. People tend to think it's brand new and it's magic, but I want to show you that a bit of history so that you can understand that many of the key ideas are actually there for a long time. So first, we're going to look at a bit of history, and then I'll talk about some common training and regularization techniques for deep learning. So, because deep learning is just another class of models. It's like linear regression. Generalized linear regression is one of the tools in your toolkit. So, like other machine learning models, it needs training, and regularization here means control of model complexity. So prevention of overfitting. So how to train a machine learning model and at the same time preventing overfitting is a very common theme for deep learning, for statistics. And third, we will look at single-cell RNA-seq data and it's the typical workflow for processing single-cell RNA-seq data. Because as I said, deep learning is one of the tools in the statistical or data science toolkit. And as computational biologists, we all have our own problems. We all have our own biological background and context. So we need to apply these tools in specific context. So as I said, single-cell RNA-seq is a good starting point. It is relatively well understood compared to single-cell ATAC-seq, for example. And as I said, we will focus on SCVI, which is an unsupervised meaning it's like PCA. It doesn't need to know class labels. It doesn't. So basically, in our unsupervised problem, we don't have Y, and it's probabilistic modeling, which means it assumes an underlying probability model for our data. So we assume our data are sampled from a certain distribution. So that's the two key characteristics of SCVI. It's unsupervised and it's probabilistic. In contrast, for example, PCA is unsupervised, but it is not probabilistic because by using PCA, you do not assume an underlying probabilistic model of your data. And last, we will talk about pros and cons of SCVI and similar approaches. So here, basically, so after this lecture, after my introduction to deep learning and SCVI, we will discuss pros and cons, because as I said, and as I repeatedly have said, deep learning is just one of the tools in your toolkit. So it, as other methods, it has its own pros and cons. So we need to know when to use it. On Thursday in the practical, we will be applying SCVI to some single-cell RNA-seq dataset. So to keep it basic, we will look at PBMC, so the simplest kind of single-cell RNA-seq data. It's simplest because the biology behind it is relatively well understood. We will see that on Thursday. So I found a paragraph from the very classic textbook, The Elements of Statistical Learning. So this is a very classic reference for statisticians. So the three authors are all well-known statisticians. So it's about, so the book is called Statistical Learning, but it's actually statistical machine learning. So basically it's a book about machine learning from the perspective of statisticians. It's a very good book. It's a bit old. The latest version was published in 2009, but still you can the good thing about this book is that it focuses on not only on methods, but also on the nature of those methods, the connection between different methods. So it's a very good starting point for understanding a number of different methods. And it has, as early as 2009, it already had a chapter devoted to, it's not called deep learning, but neural networks. Okay, because as I said, deep learning is a relatively new term. It was not such a big thing when the book ESL was published. So they just call that chapter neural networks. And in that chapter, there is a paragraph talking about history of deep learning prior to deep learning. So there are a few key time points. First, the motivation for neural networks dates back to McCulloch and Pitts, so in the 1940s. And so after its the publication of the original paper, it became a topical as today. But then a second key time point is the resurgence of neural networks in the mid-1980s. So the popularity went down a bit, and it was revived mid-1980s. And the second resurgence, of course, is deep learning. So neural networks as a class of methods, a class of algorithms, have existed for 80 years, more than 80 years. And it has its ups and downs. And every up and down is related to progress or decline in, not decline, but it's related to computational power. Because deep learning is or neural networks are a kind of computational intensive methods. So like other computational intensive methods, the popularity of such methods are always based on computational power, the progress, advances in computation. For example, if you look at the history of Lasso was proposed in 1990s, and when Lasso was proposed, it was not very popular because people didn't know how to compute it. L1 is not obvious how to optimize, and L1 a loss function with an L1 penalty term in it. So until that problem was solved, Lasso was not had not received too much attention. And you can find similar stories for Bootstrap, for example. And here it's the same. Deep learning is a nice idea. It has a good biological motivation, but its realization really depends on computational power. And its ups and downs are always related to computational power. So the motivation for deep learning or for neural networks comes from, as I said, comes from biology. And specifically speaking, it's from our understanding of how neurons work, how our brains work. So this is the 1943 paper. It is identified as the first paper discussing neural networks. So the title is called A Logical Calculus of the Ideas Immanent in Nervous Activity. So it is a very complicated title, but essentially it's saying, so logical calculus means the rules, the mathematical rules. And nervous activity means the activity of your nervous system, of neurons. So in this paper, basically, the authors proposed a mathematical model describing the mathematical properties of our brain or neural network in our brain. And what is particularly interesting is the first sentence, because of the or none character of nervous activity. So or none in modern terminology just means 0, 1. So either you have 1 or you have 0, or none. So why do we say nervous activity is or none? That's because we found that so this is a typical a neuron. It's called it has all these branches. It looks like a tree, so that's why it's called dendrite. It means tree-like cells. And for each neuron, you have all these. So these are the inputs from left to right. These are the inputs, x1 to xn. So all these inputs are in the form of electric charges, stimulation. So the cell on the one end receives electrical inputs, and on the other end, it gives outputs. So those outputs will become inputs to other neurons. So you can imagine we have a big network of neurons, because on the one hand, we have inputs, on the other hand, we have outputs, and those outputs will become inputs for other neurons. So it forms a sort of network. And it is called network because scientists have noticed that if the signal is not strong enough, then it won't be passed on to other cells. So there is a kind of threshold. So only when the input or the summation of input is strong enough, will the so only in that case our neuron will pass on that signal to other neurons in the network. So there is and we will see that later. Mathematically speaking, we have a sort of threshold. So the first implementation, so as I said in the complex title, they call it logical calculus. That is just a mathematical model of neural networks in our brain. So that is not really an algorithm as we have today. So the first implementation of neural network is not software, but actually a hardware implementation. It's called Mark I perceptron by Frank Rosenblatt. So Frank Rosenblatt was a scientist working in US, and this is the machine. So all these complex wires and are there to mimic neural networks, connection of neurons. So, and the idea comes from the 1943 paper, but if you look at the abstract of Rosenblatt's report, it says that in which the aim, so in the current report, the aim has been to formulate a brain analog useful in analysis. So here already our focus is not really on how to characterize our brain, because the 1943 paper is on that, how to characterize the mathematical properties of our brain. But in Rosenblatt's implementation, in his perceptron, in his machine, the goal is for data analysis. Is for data analysis, namely, in other words, to make predictions. We are not satisfied with just describing our brain, but we want to use a brain-like machine to do data analysis and to do prediction. So the idea of Rosenblatt's perceptron is really simple from today's perspective. It's just a linear binary classifier, which means the goal of having this model is to classify two classes, classify 0 from 1, classify dogs from cats. It's a binary classification problem, and it's a linear, which means the decision boundary is linear. So you're trying to use a line to separate two classes. So if you look at the model is really simple. So we will use similar notations in the remaining part, in the rest of today's lecture. So I explain the notation a bit. So if W transpose X, so X is our p-dimensional input. So in the central graph here, we have 6 input. We have a 6-dimensional input, a vector of 6 numbers. So in general, we have a p-dimensional input. So that's our X. And W is just a vector is another p vector representing weights. So W transpose X is just the linear combination of the input. And if the linear combination of the input plus B is larger than 0, then the output is 1. Otherwise, the output is 0. So how is it a neural network? Because if we use a neural network graph, an N graph to represent it, then in the input layer, so we have 6 features. And we want to do a linear combination using 6 weights to compute the weighted sum of the input. And then at the output, we have we call this activation function, a G. So if this value is larger than 0, then the output is 1. So the activation function is just an indicator function in this case. And another way of looking at this is, so if we write the so if we write formula as W transpose X larger than minus B, then the minus B, this term, which is called this is called bias. This is called bias in deep learning, in neural networks. Then this means the linear combination of the features, if it's larger than a bias value, then the output is 1. Otherwise, the output is 0. So here we are really mimicking the neuron, the brain activity, which is 0, 1, or all or none, as put in the 1943 paper. So that is the perceptron, the original perceptron, a binary linear classifier. And here is just an illustration. So the decision boundary is a line. So this model is extremely simple, of course, but already we can see that we are not satisfied with just modeling the brain. We are trying to use a brain-like algorithm or a brain-like machine to process real data, because the perceptron was applied to imaging data. So the first neural network model was already applied to imaging problems. Nowadays, the most popular field for applying deep learning is still imaging processing, and that has its historical roots. So here I'm introducing another model prior to deep learning. It's called projection pursuit, and it's a purely statistical model. It has nothing to do with, so when it was proposed, it had nothing to do with image processing. But the main idea is really similar. So the main idea is to extract linear combination of features as derived features. So what do we mean by this? So perceptron can be seen as a special case of projection pursuit. And in the perceptron model, so what is the derived feature? We can view this as the linear combination of the original features as the derived feature. So if you view W, the vector of weight, as representing a direction, then here essentially what we are saying is we want to project X, the original higher dimensional feature vector, onto the direction represented by W. And then we have a number, and that number is a derived feature. So instead of the original features, we have a derived feature, which is lower dimensional, which is just one dimensional. And if this derived feature is larger than minus B, we have output 1. So that is actually the same idea. So perceptron actually shares the same idea. So, but in projection pursuit regression is actually more general, because instead of using just one derived features, we can use P derived features. And so for each derived feature, we have a different weight vector. So we project the original feature vector onto P directions, and we use those P derived features to do prediction. So that is projection pursuit, PPR. So the training of PPR is, I briefly mentioned the training of PPR because already the training of PPR is not that obvious because it's more complicated than Rosenblatt's perceptron. The complexity arises from the fact that we don't have a closed form solution for the problem because, so in a typical regression problem, this is the loss function we want to minimize. So this part is the model, is the PPR model, and this is the true y. It can be a continuous value or a discrete value, and what we want to minimize is the square distance between the predicted y and the true y. So here, this is up to now we're talking about supervised problem, but our goal is to talk about unsupervised method, and we will see that in the second half of the lecture. So this is just the mean square error, a very common loss function we minimize in statistics. So different from linear regression, because in linear regression, we also want to minimize the mean square error. But in that case, we have a closed form solution. We know beta can be expressed as matrix products of X and Y. But in this case, we don't have that. So the strategy for training this is we start from just one projection. So instead of minimizing P projections simultaneously, we start from just one projection. And we use gradient-based method to find that optimal weight vector W1. And then we fix that and we add more projections to the model until the model fitting cannot be further improved. So here I want to briefly talking about this because this is really the key, the basic idea for training deep neural network, because it can also be formulated as a regression problem, and we also want to minimize some sort of loss function, but we don't have closed form solutions, so we rely on gradient-based method. So another thing I want to talk about is PPR, projection pursuit regression, as universal approximator, because an important way of understanding neural networks is through is to view them as approximator. So PPR is, so if we use the neural network representation to illustrate PPR, then different from perceptron, because in perceptron we only have an input layer which gives you all the input features and just one output layer. So we only have two layers. But here in PPR, in addition to input and output, we have a hidden layer. So if we want to write, so here I'm giving you an example. We have 6 input features, and we have 3 projections. So P equals to 3 in the PPR model, and then output. So so when people first discovered this, it appeared very surprising, because the result says PPR can represent basically any function you want. So by simply projecting original features onto different directions, and by using activation function to give it some non-linearity, and by adding them up, you can use this process, simple process, to approximate almost any function from the input layer to the output layer. But of course, you have there are some conditions. So basically, in order to have that approximation power, we have to take the number of hidden units, which is here, p to infinity. So basically the result says if we have enough projections, then a simple model such as PBR can approximate really any function. So why do we care so much about approximating functions? Because in classification, in regression, essentially we want to build a function from the feature space to the output space. So suppose your input is image. So from all those pixels, you want to make predictions of, for example, distinguish dogs from cats based on those pixels. So mathematically speaking, you're trying to find a function from the input features to the output space. So, and this result tells you if you can afford to take more neurons and you can afford, you have powerful enough computer to train such a model, then you can, theoretically speaking, you can approximate any function you want. So, but this seems to tell us PPR itself is already powerful enough for all kinds of practical problems. But actually it's not, because, so this is a theorem about how many neurons we need to achieve good approximation. So suppose the true function we want to approximate is like this. So we have D input features. We have D features, x1 to xd. And suppose the real relationship between output and input is x1 to the power of r1, x2 times x2 to r2 until xd to rd. So a very simple class of function. So suppose the true function is like this, then how many neurons we need? So this represents the number of number of hidden units we need with just one hidden layer. So if we have just one hidden layer, then in order to approximate that, this is what we need. This is the number of hidden units we need. It's exponentially large with respect to D and Rj. Okay, because it's a product of all the R's. So if the original function is complicated, so if we have a lot of we have a large D, and if R1 to Rd are large, then that means we need exponentially many hidden units to approximate a simple function. So that is often unaffordable in practice. Instead, what happens? So now we already have a neural network representation of PBR. So an obvious extension of that is to have more hidden layers. So here we only have one, but suppose we have more hidden layers, what would happen in terms of approximation? So the second inequality tells you that. So if you take more than one hidden layer, you take k hidden layer, then the upper limit of the number of hidden neurons, hidden units you need, instead of an exponential function, you have, instead of product, you have summation, and instead of R, you have log R. So that is much, much smaller compared to the first case. So this is a very simple, this is a simplistic mathematical result, but it tells you that the benefit of having more hidden layers. So the original perceptron does not have any hidden layer. PBR has one hidden layer. And theoretically, if you have more hidden layers, then it becomes easier to approximate functions. It becomes computationally more affordable to approximate functions. So that's the theoretical motivation for deep learning. So the name deep learning, in a very general sense, just refers to using neural networks with one or with a certain number of hidden layers. So because we only have deep learning around 2010 because prior to 2010, people didn't understand why we need more hidden layers. And the reason why people didn't understand why we need more hidden layers is because we didn't have the power to compute them. So first we had the power to compute them, and then computer scientists did all sorts of experiments to show that deep neural networks work. And then we have mathematicians derived this kind of results, showing that mathematically it makes sense, because by having more, so even if one hidden layer is already powerful enough, but it's computationally unaffordable, to make it computationally more affordable, we need more hidden layers. So that's the motivation for going deeper. So why are we talking about deep learning today? So now we can finally have a look at the model for deep neural network. So now, a major class of neural network is called feed-forward multi-layer neural network, or sometimes simply called multi-layer perceptron, because perceptron is the simplest kind of neural network you can have. And in deep learning, we have multi-layer perceptron. So what we have seen is that deeper neural network requires much fewer nodes to represent complex functions. So that is to say, so what is so that is to say, so our goal is to approximate regression function or classification function, and MLP just offers us a class of functions for doing that. So what do I mean by a class of functions? For example, if you want to do classification, or an even simpler case, if you want to do regression, and you would first go to probably linear regression, because it's very simple. And what is linear regression model? It assumes that the relation between your input and your output is linear. So the output is a linear function of the input. So it gives you a class of functions to choose, because even if you fix that I want linear functions, you still have a lot of freedom. You can choose the coefficients differently. So by using linear model, you are choosing a class of linear models, linear functions. By using, for example, logistic regression, you are choosing a different kind of class of functions. And by choosing deep learning, you are choosing yet another class of functions. And this class of functions can be written as this. The first formula is very complicated, but if you look at the second one, you can see that neural network can actually be iteratively defined. So suppose we have L layers. So suppose we're talking about an L layer perceptron. Then here, suppose we use H to denote the output of the Lth layer. And so this formula says the output of the Lth layer H is a function G of the output from the previous layer. So it's really like, imagine the first graph we have seen, real neuron in your brain. It takes input from other cells, and then it does some computation and it gives its output to other neurons. So here we're doing the same, except that the whole process it's much simpler, because in the actual brain you may have like circular things going on, but here everything is feed forward. So from layer 1 to layer 2, from layer 2 to layer 3. So for layer L, the output from layer L is the result of taking the results from layer L-1 as input. And what's happening here? So we have a sigma, which is again an activation function. So sigma is a known function, not nonlinear function. So when you train a neural network, you specify what sigma is. And then inside sigma, you have WL. So this is a matrix. So what this matrix does is that it projects the output from L-1, layer L-1, to, it does some linear combination of the output from the previous layer. And so this gives you a new number of, so if we look at this graph, it's actually easier. So for example, if you look at the neuron here, the node here, then you have a linear combination of all the input features. And you compute a linear combination of these features and you pass that to sigma and you evaluate and you have an output from that neuron. And again, from all these neurons, if you view these neurons as your new derived features, then if you view these derived features as your input, then what another hidden layer does is that it takes all these derived features from a previous layer as another layer of input. And again, it does the same thing, linear combination, and pass them on to a nonlinear activation function. So you do that repeatedly. So that is what's happening, mathematically speaking, inside a deep neural network. You repeatedly do an input, linear combination, nonlinear activation, and pass that on to another layer. So that's what you do. So just a few words about activation. So activation function can be chosen. There are many choices of activation functions. So a classic choice is sigmoid. So this is the same. This is the link function we use in logistic regression. So that's why logistic regression is actually a special case of neural network, because it's just a shallow neural network using sigmoid as activation function. But as we will see later, nowadays people do not use that as activation function because it's a bit difficult to compute when you want to compute a lot of neurons. Instead, nowadays the standard selection, so people always first try ReLU, which is just a, so if you look at the function, it's max between 0 and x. So if you draw it, it's just like it's like a broken line. So you have a line, but the negative part of the line is forced to be 0. So this is just it looks like a broken line. The negative part of it just vanishes. And we have other variants of activation. There are other variants of ReLU used in different scenarios, but the most basic one is really just this. So the idea is, so why do we, so ReLU is really simple. So why do we care for having an activation function as simple as ReLU? This is because if you don't have that, because ReLU is almost, well, it is almost linear because it's piecewise linear. So if you divide it into 2 parts, then the negative part is linear and the positive part is linear, but as a whole, it's non-linear. Okay, so it's a very simple form of non-linear function, because if you do not have any non-linearity, what would happen? What would happen is that however many layers you have, you just end up with a linear combination of the original features. That's because the linear combinations of linear combinations is still just a linear combination of the original features. So if you do not have any non-linearity in your neural network, then you don't need to take so many hidden layers, because however many hidden layers, you still end up with having a linear combination of the original features. So it doesn't really make sense to do deep learning with purely linear activation functions, except in some special cases. But we don't want to talk about those. So here the idea is, by having a bit of non-linearity at every neuron, and by using many layers, and by using many hidden units, so if you look at locally, if you look at bits of the neural network, what's happening is very simple, but as a whole, it's a very powerful approximator. Okay, because we have seen that even with just one hidden layer, you can approximate almost any function you like. And by having more layers, the approximation power is increased. And locally speaking, every bit of the network is very simple, but as a whole, it's very complex. So that's the basic idea. Now, let's have a closer look at the training of deep neural networks. So again, we are facing the same problem. So we have, so suppose now for example, as an example, we have a look at, we can have a look at K-class classification problems. So instead of just one output, we have the neural network has K outputs. So each output node represents for example, the probability of corresponding to a certain class. So instead of talking about binary classification, we can be talking about many different classes, and the output of the neural network are those probabilities. We have a probability vector. So here, what we want to, the loss function, what we want to minimize, instead of square error loss, we have a cross entropy. So the meaning of the cross entropy, I will not go into details here, because this is not our focus. So if you want to know the meaning, so roughly the meaning of this loss function is so if we do a lot of correct predictions, then this value, the absolute value of this loss function, is close to zero. Is close to zero. Then, oh, sorry, if we do a lot of incorrect predictions, then the absolute value of the loss function is close to zero. And because we have a negative sign here, that means the loss function is actually very so if we do a lot of correct or incorrect, I think it's if it's close to zero, then loss function is small. Yeah. So if we do a lot of correct classification, then then this value becomes very small, because it's away from zero to the negative, to the negative direction. Is away from zero. So yeah. So it's just a continuous version of classification error, because classification error is discrete, and this measure here, this loss function here, is continuous. So what we want to do is, suppose we have n training samples, and we want to minimize the so-called empirical risk. Empirical risk is just so imagine you have classification error, you evaluate the classification error using n training samples. So basically, this is just the training error. So the goal is to minimize the training error. And so of course you would ask, the goal is not minimizing training error, because if you really minimize training error, then you would have overfitting. So the real goal is to minimize the generalization error, the prediction error. But here, suppose we want to because we will talk about different methods for controlling overfitting. So suppose our goal now is to minimize the training error. Then how do we do that? As we have seen before, even for projection pursuit, simple stuff like that, we don't have closed form solutions. So what we need is a gradient-based method. And the most basic one is called SGD, stochastic gradient descent, originally proposed by Robbins and Monrose in the 50s. So this formula is just a stochastic version of Newton's algorithm, Newton-Raphson algorithm. But instead of Newton-Raphson, so like Newton-Raphson, we have a gradient here. We have a gradient here because if you look at G, so suppose the parameter we want to choose is theta. So in deep neural network, theta is a huge vector because you have so many parameters to optimize. And G of theta is the gradient of the loss function. So the gradient of the loss function is just the partial derivatives. Okay, so if you have P, so if you have 10, so say if you have 10 parameters, then then you do 10 partial derivatives respective to all these parameters, and you end up with having a vector of length 10, but each element of the vector is a partial derivative. So that's gradient. And so here you have the gradient, and the same as in Newton-Raphson. And on the other hand, you have a step size. So here the step size is a hyperparameter. So you choose the hyperparameter. You choose the hyperparameter, and the machine computes the gradient, and the machine updates the thetas. So it updates theta from theta t to theta t plus 1. And it does this recursive updating until the algorithm converges. So the idea behind it is that suppose we have a function like this. So suppose we have a very simple convex function like this, and suppose this point is the local minimum. So how do we minimize the loss function? We start from some starting point x0, and each step we go a little bit forward. We move from x0 to x1. And how do we decide which direction to move? We move along the direction of the gradient, because the gradient always gives you the direction, the optimal direction to reduce or to increase your objective function. So if this is the contour plot of your loss function and you want to minimize it, then you do it step by step. Each step you go a straight small line segment, you go straight, but you go a little bit along the direction represented by the gradient. And hopefully by doing this repeatedly, you will end up around the true minimum. So, but this, of course, is just a very simple case. To look at a slightly more complicated case, there is a very good. So there is a very good animation. So here, this is a more complex minimization problem. So still, we are looking for minimum. So the warm colors here represent high values, and the blue color here, the deep blue color here means, so this is the true global minimum, and this is a local minimum. So all these red points represent different initial values. So you can see that from some initial starting points, if we do gradient descent, then we end up at local minimums. But if we start from, for example, from here, then we end up at the true global minimum. So this is a very good illustration because it tells us first, in real optimization problems, we often have many local minimums. And also it tells us the starting values matters because some starting points are particularly bad. So if you start from those starting values, the initial values, then you will probably end up at some local minimum point. And interestingly, this graph also shows us other phenomena commonly seen in optimization, because you can see that here, this is not a local minimum. This is a saddle point. This is a saddle point. And similarly, what's happening here? So some initial values are so bad that you don't even end up at local minimum. You end up in some valley, in some saddle points. So this is keep this graph in mind, because this is actually related to what we're going to talk about in the optimization for deep learning. So that's the basic idea of stochastic gradient descent. And so the key idea is really, so we have seen two key things here. So first is the computation of gradient. The other thing is we have a hyperparameter called step size. And if you look at textbooks on deep learning, they will tell you that the most important hyperparameter is the step size. And that is because step size determines the quality of optimization. The idea is that sometimes if the gradient is very steep, so if the optimization problem, if the absolute value of the gradient is large, then you want to take smaller step size, because if your step size is large, then you may go out of the the true minimum. So because if the gradient, the absolute value of the gradient is very large, and if your step size is still very large, then that means your algorithm will not converge. It will go around and around the minimum. So ideally, when your gradient is large, you want to take smaller steps. When your gradient is small, you want to take larger steps because you want to go faster. Basically, you want to go faster to the minimum areas. So that's why if you look at the original Newton-Raphson algorithm, the step size is not chosen by hand. It is determined by the second derivative, because the second derivative reflects the landscape. So how steep is the mountains? How steep is the valley? But here, but the reason why we don't want to compute second derivative is because that's computationally expensive. So in practice, we can only afford to compute the first derivative and to use an automatic step size. So the tuning of step size is very important, because although theoretically neural network can approximate any function you want, but in practice, the behavior of your model depends heavily on optimization. How well you have you really achieved optimization? So here I want to talk about different types of stochastic gradient descent. That's because if you look at the original stochastic gradient descent, that's not what's used in modern day deep learning models. because I can tell you why. First, because if you look at the way it computes the gradient, then at each step, it uses only one data point, one x and one y, to compute the loss function. Okay, so that's again to save computation time. But the idea is ideally you would want to use all data to compute your training error, training loss function, and then you want to compute the gradient of that. But here in the original SGD, stochastic gradient descent, at each step we randomly sample one data point, one sample from the training set to evaluate the loss function and to calculate the gradient. So an obvious problem with that is we have too much variance because you cannot reliably calculate the gradient based on just one sample. So what we want to do, an immediate extension. So the first one, so in the first slot, you can see, so this is the original Robbins-Monroe algorithm. And an immediate extension for that is to use so-called mini-batch stochastic gradient descent. So the idea of mini-batch is that at each step, when you want to update your theta, update your neural network, instead of using one sample, you use a small batch of samples, say 100 samples. So you randomly sample 100 samples from your training data set, and you evaluate your loss function on that, and you calculate your gradient on that. So that reduces the variance for calculating. That slightly increased, that of course increases the computational cost, but for modern computers, it's still very fast. So now nowadays we can afford to sample a small proportion of data points to evaluate the gradient. So, so based on that, we can have further extensions. So another approach is called momentum-based SGD. So the idea is that here we still have a lot of variability because mini-batch means small batch. So the original data set can have, for example, 1 million samples, but each step you only take 50, 100 of them. So compared to the whole data set, the mini-batch is still, sometimes it's still too small. So the momentum-based idea is that you smooth the gradient. You smooth the gradient. So because in the mini-batch approach, at each step, you independently sample some of the samples to calculate your gradient. But here in the momentum-based method, if you look at this line here, so this is Vt equals to rho Vt minus 1 plus G theta t. So G theta t is just the gradient of the loss function evaluated at theta t. So here in the update function, in the recursive function, instead of having gradient here, you have a smooth version of gradient. Because if you look at the expression for Vt, Vt is a combination of all the past gradients. So if you have t steps here, then Vt is a linear combination of all the past gradients. But you have an weight term, exponential weight term here. So Vt is, although it's a result of combining all the past gradients, but if a gradient is too far away, so if suppose now I'm at the 100th step, then the gradient calculated at the first step will receive a very small weight. That's because rho is less than 1, and if t minus j, if 100 minus 1 equals 99, so if rho equals, for example, 0.9, 0.9 to the power of 99 is very close to 0. So that means I take into account gradients calculated in past steps, but I give them a exponentially decreasing weight. Okay, so by doing this, each time when I calculate gradients is not independent from previous calculations. So by doing this, I can make the calculation of gradients more stable. So all we are trying to do here is to achieve a balance, because ideally we want to use all data to calculate the gradient, but that's too expensive. So we try to use only a small part of our data to calculate the gradient, but that's not very stable. So in between, we can find a way which is still computationally fast, but it stabilizes the calculation of the gradient. And on top of that, another thing we can do is adaptive learning rate. As I said, the reason why we want to adjust learning rate is because sometimes we want if we are really close to the local minimum already, then we want to use small step size, because we don't want to go too far away from the local minimum. But if we are still very far away from the true minimum, then we want to take larger step size because we want to go faster. So that's the idea of adaptive learning rate. The most adaptive learning rate is, of course, as I said, Newton-Raphson. We really calculate the optimal way of doing adaptive learning rate is to calculate the second derivatives, but that is expensive. So we have other ways of doing that approximately. We can, so instead of calculating the second derivative, because in order to calculate so if you have 10 parameters, one loss function, calculating the first derivative means you do 10 partial derivatives. But if you want to calculate the second derivative, in the multivariate case, if you have 10 variables, and if you want to calculate second derivative, you have to calculate 10 times 10, 100 second derivatives, because you have 100 ways of calculating second derivatives. So that is too much, especially in high dimensional problems where you have, for example, in single cell, you have 20,000 features. That means you need to calculate 20,000 times 20,000 features, second derivatives. That's almost impossible. And you have to do matrix inversion, calculate the inverse matrix. That's even more problematic. So instead of doing that, we have some clever ways to approximate the second derivative, and that's how we achieve adaptive learning rate. So I won't go into too much details here, because it's enough to understand the basic idea. And then after so much talking, what I really want to talk about is ADAM. ADAM is a stochastic gradient descent method published in 2015. And this method is very, very good, very popular, because it's a combination of the moment. So first is mini batch, and then it uses a momentum-based method to smooth the gradients. And on top of that, you have adaptive learning rates. So you adjust the step size, the learning rate, according to some simpler calculation of second derivative. So all those features make ADAM computationally affordable and very stable. So that's also the optimization algorithm implemented in SCVI, for example. That's a very standard optimizer to use for deep learning nowadays. So as you can see, I spent a lot of time talking about the details behind the optimization, details behind the method. That's because deep learning is complicated. It is a blend of many things. So even if you want to try it yourself, because as we will see on Thursday in the practical, the function is easy to use. You don't even to specify any hyperparameter. You could just use the default values. But one set of default values cannot be good for all problems, of course. You have to, at some point, you have to adjust those parameters. But in order to adjust those parameters, you have to understand what those parameters mean. For example, you can adjust the number of layers. I have told you the number of layers. Sometimes by having more hidden layers, we can have fewer hidden units. And now I'm telling you, by using Adam, we can often achieve faster convergence, better optimization. So I've also talked about the meaning of step size or learning rate. And in SCBR, for example, you can adjust the learning rate. If you just want to use the default values, then you don't need to know any of these. It just gives you some result. But suppose you want to make some adjustments, and making adjustment is absolutely necessary for deep learning, because it's a very complex model. Even if you change the parameters a little bit, it may have completely different behavior. So you need to fine-tune those parameters, and in order to do that, you need to understand the meaning of those parameters. So we have talked about optimization. We have talked about deep neural network, and now I want to I'll just briefly mention backpropagation, because to go into the details is too time consuming. So we have talked about stochastic gradient descent and different versions of stochastic gradient descent. But a key problem here is we haven't really talked about how to calculate gradient. We have talked about using how many samples to calculate gradient. Use one or a mini batch, but we haven't really talked about how to calculate gradient, because the idea is typically for a complex function, it's very expensive, computationally expensive to calculate the gradients. The more complex the function is, the more expensive it is to calculate its gradient. But the good thing about neural network is that although it can be really, really complicated, because if you look at the formula a few pages ago, it's linear function inside nonlinear activation function, and inside you have another layer of activation, linear combination, etc., etc. So although it is very complicated, but calculating gradients for this kind of composite functions can be surprisingly simple. That's because, intuitively speaking, although the whole network can be very deep and can be very wide. Wide means you have a lot of hidden units at each layer. So although if you look at the whole network, it can be very complicated. But locally speaking, if you look at just a small part of it, it's very simple, because it's a linear combination, and if you use ReLU, that's a piecewise linear activation function. So it turns out that if you want to calculate the gradients for this kind of function, then you can also make use of that. So the idea is, in order to evaluate the gradient of your loss function, you go backwards, because when you want to apply your neural network to prediction, what you do is you start from the input layer, and you go one layer by one layer until you reach the output layer. But when you want to calculate the gradients, you go backwards, and you start from the output, because what you want to do is here. So you want to calculate the gradients of the loss function, and the loss function is again the function of a neural network. So you start from the last output layer and you go one layer back and one layer back again. So, and each step is very simple, because if you look just one step back, it's just linear combination and a Rayleigh function. So the idea is this structure makes the calculation of gradient easy, because you can do it step by step. And when you go through all the steps from the output to the input, then you have the gradient. So, and so this makes the calculation of gradient very simple. And the reason why people want to use GPU instead of CPU for that is because, as I mentioned, you want to calculate all these, you want to evaluate all those gradients on mini batches. And if you use GPU, because GPU is very good at parallel computing. So for CPU, if I have, say, 8 cores, then maybe for R, I have because sometimes 8 cores means 16 workers. That means I can do 16 core parallel computing. So I can divide my work into 16 parts and do them simultaneously. That's for CPU. But for GPU, you can divide your task into hundreds of parts. You can do hundreds of parallel computing at the same time. So because of the nature of deep neural network, you can divide your task into many simple subtasks. So that's why GPU is a perfect choice for training neural network. So and there are some other numerical issues, and so all these issues, if you use the SCVI function, all these numerical issues have been taken care of, so you don't need to worry about the details. But so I think I'll skip this slide because it's probably too technical. But the main idea is really, as I said, optimization and calculation of gradient, and you need to understand, so what's the role of step size and what's the role of number of hidden layers, for example. So and the last thing for the first part is a regularization. So I want to spend just a few minutes talking about regularization, because up to now what we have talked about is how to minimize training error. But as I said, if you really minimize training error, then that leads to overfitting. So how do we avoid overfitting? There are several, four main ideas, but essentially they are all related. So the first technique is called weight decay. So it's just ridge regression. So instead of minimizing the original training error, you try to minimize training error plus a penalty term, L2 penalty term. So L2 penalty term, if you are familiar with the ridge regression, that means you don't want your the sum of the squares of your parameters to be too large. So it is a way of controlling the model complexity. So like in ridge regression, because in linear regression you have a similar problem. If you have a very large model with many for example if you have say 100 samples, but you have say 80 features, then that often leads to overfitting. So in that case, you want one solution is to add a penalty term, L2 regularization term, that sometimes people prefer not to do that, because if you add a regularization term, that makes the optimization problem slightly more complicated. So instead of minimizing just the training error, now you need to minimize a slightly different, more complicated loss function. So there are easier ways of avoiding that. One is called dropout. The idea of dropout is that each time when you do the gradient descent, because the way you do gradient descent is that you divide your data sets, big data sets, into a lot of mini batches, and you use those mini batches, and each time you use one mini batch to update your parameter, and then you use a different mini batch. So if you divide your large data sets into, say, 1,000 mini batch, then after 1,000 iterations, you have used all the samples, and you can do it again by dividing the training set in randomly into different mini batches. So you can do it again and again, for however many times you like. But the idea of dropout is that each time you do that iteration, you train a slightly different, you change your neural network, slightly change your neural network. So the idea is you randomly drop a number of neurons or number of internal nodes from your neural network, so that each time you are training a slightly different neural network. So why do we want to do this? Because this prevents overfitting, because first you can prove that in some simple case, this is equivalent to L2 regularization. So this means by simply randomly dropping some internal neurons, some hidden neurons, hidden units, you are still minimizing the same loss function. Well, you are still minimizing the training error, but effectively you are also your loss function has an L2 regularization term in it. So that means you don't need to explicitly add an L2 term into your loss function. It is all done implicitly. That is great, because that means you can use the same old algorithm, but now you have an L2 penalty term in your loss function, and you don't have to really worry about how to minimize the L2 penalty term. So that is because mathematically, randomly dropping some nodes in some cases is equivalent to doing L2 regularization. And there are some other methods, but essentially, so theoretically, you can show that many of these methods are actually equivalent to, in some simple cases, are actually equivalent to L2 regularization. And so that's great, because by doing simple things, we achieve similar effects as more complex methods. So that's why people prefer, for example, dropout is implemented in CBI because it's easy to implement, and you can also implement early stopping in SCBI. So early stopping means instead of just looking at the training error, we divide the data into training and validation. So we calculate. So at each iteration, we also calculate the validation error, because the validation set is not in your training set, so it is more objective than the training error. So early stopping means if your validation error does not reduce for some time, you stop the training process. You stop the updating of your neural networks, of the parameters. So, okay, so all these are technical sides of deep learning. What about using it, apply it? What about applying deep learning to biology, to single cell RNA sequencing? So the next part we will be focusing on SCBI, and my plan is I will focus on the model of SCBI, because as I said, if you want to use SCBI, you really need to adjust the parameters. And I have from the first part, we have discussed meanings of different parameters. The learning rate sometimes is also called step size, and the role of number of neurons and number of hidden layers, etc. But SCBI is actually more complicated than that, because it's based on a probabilistic model. So here, in order to understand SCBI, you have to understand that underlying model, the logic behind the neural network. So the motivation for SCBI is that, because typically in a workflow, in a single-cell RNA sequencing analysis workflow, for example, if you look at this paper by Lucan and Theus, it's called Current Best Practices in Single-Cell RNA-seq Analysis. So this tells you, because a typical workflow has many different steps, and this paper gives you recommendations for each step what to do. So typically we start from a count matrix, where the rows are genes and the columns are cells. So it's a bit different from the machine learning convention, because typically in machine learning we have samples for rows and features for columns. But we start from count matrices, and the counts are observed counts of mRNA. So that's the starting point. And the second step is usually quality control, because we have a lot of cells that aren't expressed, that we have a lot of cells that do not contain many gene expression counts. And we also have some genes, many genes that are not really actively expressed in cells. So we want to delete those. And the third step is usually normalization and feature selection. We want to do normalization because the library size in single-cell study, as in bulk RNA-seq, library size is usually unwanted. Because many people tend to, in many data sets, the reason why you have different library sizes is because it's purely technical. It's due to technical reasons. So we don't want to. So normalization, if you look at normalization methods, many methods focus on how to get rid of the correlation between, get rid of library size, because it doesn't reflect biology. And feature selection here means sometimes people take, typically in a single cell RNA count matrix, you have 20,000 features, 20,000 genes. That's whole genome, because we as human, we only have 25,000 genes. So, typically in one study, only 1,000 to 5,000 genes are used. Some people only take 1,000 to 5,000 genes. And what they do is by taking the most highly variable genes. So they filter out majority of the genes and they normalize the data. And data correction refers to batch effect removal. So your data could be from multiple laboratory, from different techniques. So sometimes you want to remove batch effects. And imputation means because the data count matrix is very sparse, contains 90% of zeros, for example. So sometimes people they want to impute those zero values to make zero values non-zero. And after all those normalization, correction, and the corrected normalized count matrix is used for downstream analysis, for visualization, clustering, differential expression, or trajectory analysis. So this is a very complex workflow. And at each step, people use different tools and people make different assumptions. So the motivation for SCVR is that can we use a single model that is complex enough, powerful enough to account for all these variabilities, and are powerful enough for all these purposes. So by having a powerful model, I can use this model to do, for example, normalization, batch effects correction, or imputation and clustering. So can I have this? So now the first problem is what model to use, because when we talk about model in statistics, we're talking about a statistical model. A statistical model means represents a reasonable belief about the distribution of the data. For example, in linear regression, for example, in most testing problems, in hypothesis testing, we do all sorts of transform so that our data looks like a normal population. So, and then we use normal-based methods to do differential analysis. And so, for example, t-test. And t-test is based on normal assumption because one of the most important conditions for t-test is that your data has to be normally distributed. So, and in order to make your data normally distributed, often you have to do some transforms. So when we talk about model, we're talking about something like normal model for t-test. So now we're talking about single-cell RNA-seq count matrix. If we want to directly model that, what model should we use? Now, people tend to agree on using either negative binomial or zero inflated negative binomial. So negative binomial model distribution can be seen as an extension of Poisson distribution. So in Poisson distribution, you only have one parameter, lambda, and lambda is the mean of your Poisson distribution, and it's also the variance of your distribution. So if you use Poisson to model your data, that means you implicitly assume that the mean of your data should be close to the variance of your data. So that is the fundamental assumption you implicitly use when you use Poisson model. But that assumption often does not hold. So if you look at, if you draw, for example, in this graph, if you draw the variance, gene variance against the gene mean, and so if the data is truly Poisson, then you should observe all those points centered around the dotted line, because the dotted line means your variance equals to your mean. So if your data is truly Poisson, you should observe all those points close to the dotted line. But instead, you can see a shift upwards. That means the gene variance is significantly larger than gene mean. So, and that is called over-dispersion in biostatistics and in statistics, of course. So that assumption is violated. So Poisson is for counts, for integers, but is too strict to use because it assumes that your mean equals your variance. So in order to avoid that, you can use negative binomial, because in negative binomial you have two parameters, so you can have larger variance than mean. And the reason why people sometimes also want to use zero inflated negative binomial is because there are too many zeros. So if you just fit a negative binomial model to your data, then it underestimates the number of zeros. It cannot account for the excessive number of zeros in your data. So if that's the case for your data, then you probably need to use zero inflated negative binomial model. So zero inflated negative binomial distribution has 3 parameters, 2 from negative binomial. So using those 2 parameters, you can account for mean and potentially different variance. And a third additional variable is for zero inflation, is the probability of getting zeros, for example. So so from Poisson to negative binomial to zero inflated negative binomial, we have an increase of model complexity. We have 1 parameter to 2 parameters, 3 parameters. So why do these assumptions matter? Because using the correct model for your data is very, very important for normalization. So if you, so for example, if your data has a lot of dropouts, have excessive number of zeros, but you use negative binomial, then the model fitting will be distorted. So the model fitting cannot be very good because it underestimates the number of zeros, and that may affect your downstream analysis. Because, for example, if you use HR or DE-SIG2 for differential expression analysis, those differential analysis are based on essentially hypothesis testing based on negative binomial assumptions. But if the negative binomial assumption is wrong, then the testing result will be inaccurate, which means the biomarkers, the marker genes you derive from those testing won't be very reliable. So using the correct assumption is very important. And how do you know whether you are using the correct assumption, the correct model? By looking at your data, by looking at a series of diagnosis plots. So, so as I said, choosing a correct model is very important. And the motivation for SCVI is to use the same model for everything. And so the base, SCVI is the model of SCVI is written here. It's it looks very complicated, but we will break it down into 3 parts, and you will see that it's actually not that complicated. So the idea is we use zero inflated negative binomial. So we have a negative binomial part here, and we have a zero inflation part here in our model. So let's go through it step by step. So I think this page is the most important page for the slide for the whole lecture, because it's about the underlying model for SCVI for single-cell RNA-seq. And so first, let's look at the notation. So suppose we have observed counts from a count matrix, and we denote it by X and G. So N is the index for cells. So we suppose we have capital N cells, and small lowercase n, small n is for the representing is the index for cells, and G is the index for genes. So suppose we have a big N times big G matrix, and each element in the matrix is a count, is X and G, small n, small g. And we use a cell and gene specific zero inflated negative binomial distribution, which means for each count, for each number, we fit a distribution. Now, that is definitely, by itself, that is impossible, because if you have n data, n times g data, if you have n data points and g features, you cannot fit that many models, because here we're talking about big N times big G models. That's impossible. We have too few data for that. But the idea is not really So the idea is that all those models are not independent. All those models are actually related. So in fact, we don't need to fit all those models independently. So but before that, let's look at some other notations. So P, X, and G is the probability mass function. The probability function for is the distribution function for zero inflated negative binomial distribution. And if you look at the right hand side of in this bracket, what we have is we have Sn, and Sn is the batch index. So Sn means for sample n or for cell n, which batch this is the batch index for that cell. So all these cells may be from different batches. So Sn is the index denoting batches, and it is known because you know which cells are from which batch. And Ln is the log library size. So as we said before, the reason why we want to explicitly model library size is because normalization, because it is something we don't want. So as I said, in bulk RNA, in single cell RNA analysis, we often don't want the variation caused by different library sizes. So here we explicitly model library size in order to get rid of it. And that's also why we want to explicitly model Sn, the batch index, because we don't want the variation caused by batch index. That's batch effects. So we want to remove batch effects, we want to remove library size. So that's why we explicitly model them in our model. And does this mean we have so many parameters to estimate? Because for each zero inflated negative binomial, we need 3 parameters. And we have n times g negative binomial models. So does this mean we really need to estimate so many parameters? The answer is of course no, because we are using a kind of latent model, because we have the basic assumption is that the distribution of xn, so here xn is a vector, so this is the gene expression levels of cell n. So it has g, it's a g vector because it has g genes. So the idea is that the gene expression of the same cell is determined by the same low-dimensional latent vector, denoted as Zn. So for each cell, there is a unknown latent vector, lower dimensional vector behind it, and all the variation of the cell gene expression in that cell comes from those latent vectors. And in SCVI, the default number for latent the number of latent variables is 10. So that means even if you have 20,000 genes, you model them by 10 numbers. So the real dimension of your problems is not G, is not 20,000, but 10. So that is the idea of latent modeling, because the information is sparse, not all dimensions are useful. So you assume that all the information comes actually only 10 features, 10 hidden features. So now let's look at the hierarchical model. So it's a Bayesian model because, as I said, if you want to use the same model to model everything, then you need to go to Bayesian model, because the biggest difference between Bayesian and frequentist modeling is that for frequentist modeling, you divide your tasks into different steps, and for each step you use a potentially different model. But in Bayesian modeling, you really want to universal model, the same model, to account for everything. that is a simplistic summary of Bayesian vs frequency modeling. So the motivation for SCBI is really Bayesian. So in Bayesian modeling, of course, we have prior and posteriors. So in Bayesian models, we assume that all parameters are actually random variables, like your data. Your data are random variables, and the hyperparameters are also random variables. And prior to your data, you assume some prior distribution for those parameters. And then after you have data, you use those data to update your belief about the parameters. So prior distribution represent the prior belief for the parameters, and posterior distribution represents the updated belief after you have taken data into consideration. So prior distributions, so we want to model Zn and Ln. So prior distribution, we assume Zn follows a normal distribution, zero mean, and the identity matrix. So this is just a common way of specifying prior distribution. This is just saying we don't know anything about latent vector when we haven't seen any data. So we just use a common standard normal distribution as the prior distribution. And Ln, the prior distribution for Ln follows log normal. That is commonly used. Log normal is commonly used for modeling library size, log library size. So L mu, so the mean and variance of log normal is actually estimated from your data. So in SCBI, those two parameters are actually estimated from your data. So this is the mean library size, log library size, and this is the variance of the log library size. And so by doing this, if you are familiar with Bayesian method, you would immediately think that's not Bayesian because in the most standard Bayesian analysis, you cannot use data to estimate your prior. But here we are doing some kind of empirical Bayes, so we just estimate some of the parameters from the of prior distribution from the data. That's for simplifying the problem. That's for simplification. So we have some prior distributions for Zn and Ln, and now this is the key part. We have rho n, which is a vector for which is from I should write. So this should be equal. Okay, this is not. This shouldn't be tilde. This should be equal. So rho n is so FW here is a neural network. Okay, so we're talking about our model, but you can see why SCBI is a deep learning based method, because a part of the model is actually determined by neural networks. So here rho n is the output, is a vector, is the output, is a G vector, and it's an output from a neural network. So what's the meaning of rho n? As I said, rho n is a G vector. So rho n G, which means the Gth component of the rho vector. So the x tilde n G, so x tilde is the normalized. Sorry, it's not normalized. So suppose we don't have zero inflation, then x and g follows a negative binomial distribution where the first parameter is Ln, rho n G. So Ln is the log library size, and rho n G. So the meaning of rho n G is some kind of proportion or weight for distributing so the idea is the summation of the rows from rho n1 to rho ng. The summation of all those rows equals to 1. So it's like a probability vector. So the idea of this is that you are so you have some log library size, and you want to distribute the log library size to all genes. Okay, so the parameter rho n is very important, because that is used as the as you will see on Thursday, that is used as the output. So rho n is some kind of normalized gene expression data. So after normalization, instead of xn, you have rho n, and rho n is a vector summing up to. So if you sum up all the elements of that vector, it sums up to 1. So it's like so another case where you can have that is you use CPM count per million or normalization, where you simply divide all the counts by library sizes. So in that case, you have a vector of proportions. But here, this is just a more involved version, a more complex version of doing that. So the output, the normalized vectors, is still a vector of proportions, but it's an output from a neural network. And if you look at the input of the neural network, you have latent variable Zn, and you have batch index Sn. So the meaning of that is, so how is the data how is the data generated? So in order to have so suppose we want to sample from this model, what we do is we sample Zn, the latent vector, from the latent vector distribution. And when we have a Zn, we choose a batch index Sn, and we put this into the neural network, and the neural network gives us a vector of sort of proportions. Okay, and then we put that vector into negative binomial distribution, and in the negative binomial distribution, we have log library size, log library size. And what we have is a negative binomially distributed xng tilde. Why? So the reason why we write xng tilde here is because we haven't accounted for zero inflation. So if you want to model zero inflation, this step is optional. So if you want to model zero inflation, then the model is slightly more complicated, because if you look at hn, so if hng equals 0, then the xng is simply xng tilde, otherwise it's 0. So basically hng controls whether the value is dropped out or not. So here we are talking about how the data are generated, and this is this is how we model the data. So first we have a latent variable, we have a batch index, and then we have a negatively binomial distributed count. And up to now, we don't have dropout. And how does dropout happens? It is determined by another neural network, FH, which also takes Zn and Sn as input. So this neural network accounts for dropout. So this part, the last two equations, is modeling zero inflation. So with this 6 lines, it gives us a Bayesian hierarchical model. So first, we assume we have the gene expression of cell N is governed by 10-dimensional latent vector. And the 10-dimensional latent vector is normally distributed. So first we sample a latent vector from the normal distribution, and we sample log library size from the log normal distribution, and then we put those if we put Zn and Sn into a neural network, we get the normalized data. So the normalized data is the data without noise. And then where does the noise come from? First, we need to put rho n into MB. So that step gives you influence of log library size. And then we put X and G tilde into a zero inflation model, and we have zero inflation. So that's a probabilistic model. A probabilistic model is the reverse of a statistical model, because a probabilistic model tells you how the data are generated. Of course, it's a hypothetical model. It's not real, because we never know how data are generated, but we just use this process to model what's there, what's observed. So these few lines give you a data generating process. First, you have, because in real life, what you do is you first account for zero inflation, you separate zero inflation and the negative binomial part, and then you normalize the negative binomial part, and then you get rho, the normalized data. But in a probabilistic model, first you have the normalized data, and then you have negative binomial, and then you have zero inflated negative binomial. So that is the model. Okay, we don't have much time, so I'll probably just because we have more time on on Thursday, so I will probably let me see if I can. Yeah, we don't have much, so I'll keep. So we'll continue from here on Thursday. So the plan is that we use 30 minutes to 40 minutes to finish the model, and then we have a look at application of the SCBI. So we have already talked about the most important part of the method, that is the underlying model, but that is only the model. The problem is always, so when you have a model, you always need to you still have the problem of making inference. So because the model is just a structure, but in order to make that useful, you have to do estimation. For example, you have to estimate all those parameters, and in Bayesian statistics, this step is called Bayesian inference. It's essentially just training, model training of a Bayesian probabilistic model. And then this step is also pretty important, because here you can really see the role of neural networks. Okay, so we will continue from this page next week, and sorry, this page on Thursday. We'll use 30 minutes to finish the slides. Okay, so see you on Thursday.